Hello, thank you, Sarah, Diane, and Christine for organizing this session and for inviting me, and thank you to you all for coming to participate. Today's primer will be a conversation about different experimental tools for measuring the temporal dynamics of RNA expression. So I'll start with some background on some of the interesting questions that lie in this space, and then take you through some of the tools that have been developed, ending with our own tool, RNA timestamps. So by the end of this, hopefully you'll get an idea of why temporal tools are important, what strategies exist, and an intuition for thinking about experimental methods and RNA dynamics. Like Diane said, feel free to post any questions, and I'm happy to answer them over the course of the presentation. In my opinion, it's always better to get some clarification rather than be confused until the end. Before we get into why dynamics are important, let's just take a small step back and think about the advances of RNA sequencing. So measuring the transcriptome is a good way to begin to understand what a cell's state might be. It's a dynamic and responsive medium that the cell uses to respond to its environment, develop down a certain lineage, or respond to disease, and so on. And so as scientists, we're really excited about trying to understand how to measure these cell states. And single-cell RNA sequencing gives us the resolution to distinguish between individual cells. And so you can imagine that you have some tissue where the ground truth is that you have cells that are heterogeneous for expression in the gene. And measuring in bulk would normalize any variation in subsequent analyses. Whereas single-cell sequencing enables us to distinguish between different cell types by looking at each cell separately. So to place single-cell RNA sequencing on the axes of time versus gene expression, you get a single point of information from a single-cell RNA sequencing experiment. Of course, there are many dimensions corresponding with the many different genes that you're looking at, but each sample of cells that you take and lyse and library prep, you get single time points of each cell's transcriptome. And you can think of this as a position in time. So scientists have generated these rich data sets by designing experiments that sequence cells from multiple different samples and multiple different time points. So for example, if you wanted to look at how tumor cells in a mouse responded to a checkpoint inhibitor treatment, you might look at cells from 3 different mice. You would harvest cells from one mouse at day 0, and then another on day 2, and then on day 4, and compare what their transcriptomes look like. So this sort of study has yielded numerous findings and works well for some questions where these cohort comparisons are sufficient for what you're interested in. But it's important to note that you're measuring what you're trying to compare is the same cell at multiple different times in different samples. So there are a few roadblocks to this sort of analysis. So one, sometimes it's difficult to know when you should be sampling your experiment. So for example, here you have some ground truth change in RNA expression, and you're sampling at these 3 time points. And it's really hard to know without sort of any understanding of the biology necessarily that the most interesting changes are happening at these time points, and you should be really sampling there. And so these changes also happen really quickly in the span of minutes to hours. Sometimes you're actually unable to sample often enough to get really high time resolution. And moreover, sometimes you want to study the same cell as opposed to these different cells. And that way you can understand the trajectory of a single cell in gene expression space, or even where it came from. And there's this great paper on some of these fundamental limitations of trying to infer these RNA dynamics from single cell snapshots by Caleb Weinreb and others from the Long-Klein's group. It models this problem as you have some cells traveling through some gene expression space, shown here as this cube. And when we do single cell RNA-seq experiments, we get this sort of static snapshot of cells in transcriptome space. So you can imagine each point here is a single cell, and maybe points that are closer together have more similar transcriptomes. And so trying to infer the dynamic relationships between these cells is a little bit of a black box. Like, we know these closer points are more similar, but but what direction are these cell states maybe changing? So how you assume the entry and exit points of the gene expression states, or maybe the start and end points of the trajectory shown here is these pluses and minuses, can change how we infer the direction of the gene expression changes. So if you think the start of this trajectory is maybe in the middle, and it moves to these exit points in the end, you would think that the direction is sort of going outwards from the middle. And depending on where you assign these points, what you think about the biology might be different. It's also hard to figure out how much of what you're seeing is just random stochasticity. So you might have cells that are in very similar cell states, but actually heading in totally different directions in their trajectories. You can imagine some cells oscillate between states or have some hidden cell states that aren't encoded in RNA that determine the dynamics of their RNA and their future cell state. So if we want to get an idea of RNA velocity and dynamics, we need to look at different methods that give us an idea of velocity at each position. And so measuring velocity, so you have maybe one cell that you're measuring, but you can predict a future state or infer a previous state, gives us a better idea of the immediate changes in gene expression given a perturbation, and enables us to predict how expression might change in the future and so on. Hopefully you can see where I'm going with this analogy, and that naturally there are biological questions that necessitate measuring another derivative of time, or acceleration. And while the methods for RNA velocity enable us to compare two time points for the same cell, often we want to understand the history of RNA expression. So you can imagine some cell that had some complex history, and we want to understand the program that led to this cell being where it is in this current moment in time. And so our lab's method, RNA timestamps, tries to tackle this problem by using adenosine deaminases to make edits over time, which we can use as a proxy for the age of each individual transcribed mRNA. And I'll describe that idea and our method towards the end of this primer. So that marks the end of this conceptual background. Are there any questions that I can help to answer? And if not, I'll proceed.
Thanks, Sophia. I don't see any in the Q&A, but I'm sure some will come up later. So,
okay, great. So the first method, RNA velocity, it was developed in single cells by Joelle Lamano and many others from Stan Linnerson's and Peter Tarshenko's groups. And it builds off of this idea that was actually initially performed on bulk samples and can predict the future state of cells on the timescale of hours. So basically you can measure the amount of unspliced versus spliced mRNA to estimate the rates of splicing and degradation and infer the dynamics of the entire transcriptome. So as many of you know, DNA is composed of introns and exons, and mRNA is the result of RNA transcription, intron splicing, and post-transcriptional modifications, such as 5' capping and 3' polyadenylation. And when you do single-cell RNA sequencing, you typically use oligo DT primers to enrich for mRNA, which has that 3' poly A tail. And so in your sequencing results, you would expect to see only exons. The authors found, however, that between 17 and 23% of the reads, depending on which method you used, contained unspliced intronic regions, which is because they thought there were these secondary poly T priming sites in introns. And just as in terms of definitions, they defined any spliced reads as reads that matched only exonic regions, and unspliced as reads that contained any portion of an intronic region. And so you can see here that they built this simple model for splicing. This DSDT term, or the rate of change of the spliced mRNA, is what gives us velocity information of each gene. So assuming a splicing rate of 1, the rate of change of the spliced RNA is the difference between the rate of cell that is making spliced mRNA times the amount of unspliced mRNA, minus gamma, the rate of degradation, times the amount of spliced mRNA. So at steady state, when you have constant transcription, and no change in your spliced RNA, so DSDT equals 0, you can think about this as U equals gamma S. And this gamma can be considered a sort of slope that contains all of the degradation and splicing rate information and any other variables like intron and exon lengths. So interestingly, they found that most genes actually had consistent gammas in different cell types, but 11% of genes showed different slopes in different tissues. So first, you would estimate the gamma by regressing the expression of many cells, and then you can determine when the cell is not in steady state. So stepping back a bit, you can think of RNA dynamics more broadly as if a cell is trying to increase its future production of a certain gene, you should see an increase in unspliced mRNA and vice versa. And at equilibrium, there's a certain amount of unspliced mRNA that you would expect given how much spliced mRNA that you have. And in a dynamic state, the amount of unspliced mRNA U will be more or less than what you would expect given your slope gamma. So here you can see these two different phases, induction, where the unspliced is greater than what you would expect, and repression, where the unspliced is less than what you would expect. So as a proof of concept, they look at these circadian rhythm genes in bulk in the mouse liver, which have a known direction that matches with time, as you can see here. And they found that the unspliced mRNA levels at each time point, which corresponds to this U here, were more similar to the spliced in the next time point. And so here are these two genes, CDS and FGF1, in the same sort of phase portrait as earlier. You can see the induction and the repression phases, and that matches well with time as their circadian rhythm genes. On the scale of the cell state, however, we want to understand what direction the cells are moving in and what their next state will be. And you can imagine if you were to do this experiment with a single cell, you would take all of the cells and maybe plot the first two principal components of their gene expression in space. And you get lots of points in the space with no understanding of how they're related, except maybe that closer cells are similar in that space and their transcriptomes. And so here, each circle is what they observe, the current state, and the arrow is pointing to the expected direction of this future state. And they solved the predicted future state by solving these differential equations for every single gene. And using these arrows, you can see that the direction that they're able to infer from the spliced and unspliced data matches the expected direction of the circadian cycle and time, which is awesome. And so I think this bulk analysis is really interesting, but the really powerful thing about RNA velocity is that it builds upon single cell techniques to bring RNA dynamics to the single cell level and can identify future cell states in contexts that may be more complicated than circadian cycle. And so this is one example, and I'll explain this. It's a beautiful single cell application using RNA velocity. And here they looked at single cell data in the developing mouse hippocampus. So this is a t-SNE plot that visualizes all the cell's transcriptomes in 2D space, where cells with similar transcriptomes are locally closer together. And they looked at markers to determine what the cell types were at the ends of these branches. And so you can use their spliced and unspliced data to project a vector field onto this t-SNE and identify which direction development is occurring in. And the arrows here are sort of locally averaged because there are so many points, so showing them all might be a little bit tough. And you can use this vector field to simulate using a random walk, run in forward and in reverse, using these velocities as transition probabilities to identify where the root and the endpoints are, which you can see here. So this is sort of the beginning point, and these are the endpoints given these RNA velocities. And so you can orient this lineage without any understanding, any prior knowledge about what hippocampal development looks like. They also show that RNA velocity is able to work in the human embryo later in their paper. which is an example of a situation where sampling frequently may be difficult. Overall, I think RNA Velocity is a great tool to use in your existing RNA-seq data sets that can predict cell dynamics from real transcriptional kinetics. I'm not an expert on this method, so I see a question about it. I can try to talk about it. So the question is, how many cells per UMI are they using to make this TSNE? I'm not sure. I assume it's UMIs per cell, but they do show it in 3 different standard standard transcriptomic methods. I think they show it in SmartSeq2, InDrop, and 10x, and it seems to work well in these different platforms. They also have, I think, a package online that you can access to sort of just put in your own single cell data to see how it would, the inference would look. So I guess as useful as looking at spliced and unspliced RNA are, there's a caveat to looking at introns. They're pretty sparse and have relatively short half-lives. So you're confined to studying endogenous RNA dynamics. And having an experimental method to do a pulse chase to measure newly synthesized mRNA would increase our control over the temporal dynamics that we're interested in studying. So just a quick refresher on how pulse chase questions work, or pulse chase experiments work. In general, there are these 2-phase techniques that are used to examine cellular processes that take place over time. So the classic example is the Meselson-Stahl experiment, where they determined the mechanism for replication of DNA. And they grew bacteria in heavy nitrogen, N15, which was the pulse, and then chased with N14. And then they purified DNA from the bacteria and put it in a cesium chloride gradient, and then compared what they saw to standards that you've measured where all of the DNA has N14 or N15. And based on the bands that you see after many generations, you can determine that replication is semi-conservative of your 3 hypotheses. And so you can imagine doing a similar thing with RNA, where you add a compound that incorporates over time as a pulse that can be measured later.
Sophia, there is a question in the Q&A. Unfortunately, I'm not able to see it, but I hope that you can. So.
Oh, interesting. I can see it. Okay. How to account for cell cycle stage S1 and other hidden covariates? I'll actually be talking about another method that is able to distinguish between cell cycle state and sort of different driving programs that are changing transcription. And I hope that'll start to answer your question a little bit. But the nice thing about being able to compare 2 different cohorts of newly synthesized versus sort of existing DNA is that you can suspect that newly synthesized DNA is more likely to correlate with a stimulus that you're presenting to your cells, but the leftover DNA that you can see, might show some other information, and I'll talk about that once we talk about Skyfate. But that's a great question. I think I'm sure you're interested in it, but also a lot of other people want to try to answer that question. So just before that, it's like the next thing. Talk a little about SlamC, and Veronica Herzog and others from Stefan Amariz's group made the sort of experiment where you pulse chase RNA possible on the single cell level with their improvements to 4SU labeling in SlamC. So 4SU or 4-thiouridine, or here I guess S4U, is a popular nucleotide analog that people use to study RNA expression dynamics. And so prior to Veronica's development, scientists would perform affinity-based RNA purification to isolate RNA in bulk that had incorporated the 4SU, you can imagine, in like pulse chase experiments. So doing so is laborious and has low signal to noise performance. And so extracting in a single cell and doing the purification, you probably wouldn't get a lot of RNA to do an analysis. So at SlamC, they developed this orthogonal chemistry approach And they use this thiol-linked alkylation step, which converts thymines to cytosine during reverse transcription. And you can imagine reading out your pulse chase experiment with sequencing and skipping the affinity-based purification entirely. So the experiment would be as follows. So at time 0, you incubate your cells with 4SU, which over time will integrate into the newly synthesized RNA after some delta T. And this chemical IAA does thiol modification that causes a carboxyamidomethyl group to be added to the 4SU. And this reaction eventually causes Gs to be incorporated instead of As, Gs instead of Ts, and then and so on. You would expect to see As and Ts. Where you would expect to see As and Ts, you would actually get Cs and Gs in the cDNA. So since their development, there have been many methods that have used 4SU labeling to look at RNA dynamics in single cells. And the most scalable method is Guydate, a method developed by Zheng Cao and several others in the labs of Cole Chapnell and Jay Shingiri. It combines this idea of using 4SU labeling with single cell combinatorial indexing, RNA sequencing, to give insight into the dynamics in short time windows of 1 to several hours. So like before, these cells are incubated with 4SU to label this newly transcribed mRNA. And an important step that they do is when you're harvesting your cells at T1, you fix them. And that way you can perform this chemical treatment in bulk rather than having to have your cells sorted and then extracting the RNA individually and then performing this kind of chemical modification. And then you can dilute them into these 96-well plates, index one side, and then pull the cells again, and then randomly sort into a new 96-well plate for the other index. And doing this sort of two-step indexing is this combinatorial process that means that most of your cells have unique barcodes. And so later on in the analysis, you can measure the T to C conversion rate to understand where your newly transcribed mRNA is. So I think a really great part of this paper is the analyses they do using the dynamic information. And so they study the transcriptional dynamics of the cortical response by looking at an in vitro model of adenocarcinoma-derived cells. So dexamethasone is this synthetic form of cortisol, and it activates the glucocorticoid receptor and quickly affects gene expression. And they look at multiple time points after presentation with DEX, incubating the cells for 2 hours prior to harvesting with 4SU. So as an aside, they found consistently that about 20% of the mRNA was labeled with the 4SU, and intronic reads had higher amounts of labeling, which makes sense because we would expect that intronic reads are more likely to be more recently synthesized. And they sequenced over 6,000 cells this way, and here's a plot of them in 2D using UMAP. So these 3 UMAPs show how looking at newly synthesized RNA versus the whole RNA gives you different information. In the whole RNA UMAP, you don't really see a clear separation between the no treatment condition of red and the 2-hour condition in yellow. If you only look at the newly synthesized RNA, however, the untreated condition is distinct, which demonstrates that the newly synthesized RNA is probably more representative of the DEX response. They actually found that the fold changes associated with the response had a much larger magnitude, looking at the newly synthesized RNA rather than the whole RNA. And this last UMAP uses joint information from both of these by taking the top principal components. And you can see it's able to separate 0, 2, and 4+. So these plots down here are the same UMAP structures as sort of the ones that you can see here pointed by the arrow, the whole RNA and the joint information. And here that this UMAP is colored by clustering. And you can see that one thing that changes between your a whole RNA clustering and your joint information clustering is that 1 and 4 are split into 2 different groups. So this is colored with the same clustering as shown here. And so they were looking into this, and they actually saw that this split is related to G to M cell cycle markers occurring in a portion of the cells and not in the other. You can only see this if you look at the newly synthesized RNA. Actually, you don't really see it if you don't look at this joint information plot. And then if you're interested in transcription factors, they also have another figure that talks about how they can look at which transcription factors are correlated with this newly synthesized RNA, and we're able to identify many different links between TFs and genes that were sort of previously undiscovered for the glucocorticoid response. An important finding that makes this distinct from pseudotime is that SciFate can predict future and past states from the same cell. So after estimating their detection rate and the degradation rates for each mRNA, they're able to infer transcriptional states in the past at the beginning of 4C labeling in the future. So you can imagine that you have a cell here from maybe the 2-hour time point, and you can look at the leftover transcriptome that isn't labeled and infer what an earlier cell point might be. You can call this a parent cell that matches it. And so using this method, you can connect cell trajectories across many different time points that are informed by many different measurements in time, unlike pseudotime measurements. Does SciFate work in a mouse? Oh, great question. So towards the end, they talk about how their paper is a lot of these in vitro methods, because it's hard to do 4SU labeling in a mouse. And they talk about how there's a mouse line that would be compatible with 4SU labeling. So I don't think it's a space that has been explored too much, as to my knowledge, but I think people are trying to think about how to use metabolic labeling in vivo. Okay, so for you cell cycle lovers out there, so this UMAP on the left is an example of how they connected cell trajectories between different time points. So each tier here is a time point, and this is the UMAP, right? It's just sort of visualized in 3D space. And so you can see all these like little lines, and that's the result of their work connecting the most likely previous cell state, given what you can see in your RNA, the newly transcribed versus the leftover mRNA. And so then they used these trajectories, and they were like, oh, like what if you could cluster the trajectories and see what you get? And so you can plot a new UMAP where you're clustering all these different paths, and you can see 3 different and distinct trajectories. And so as this makes sense in that each trajectory actually shows a transition from low to high glucocorticoid activation, which is, we're putting in the DEX methadone and then doing this experiment, and that's what you would want to see. But the trajectories actually correspond with a different start point in the cell cycle. So this first trajectory corresponds with cells that transition from G to M to G1, and the second is late S to G to M, and the last is G1 to S or G1 arrest. So I think a really cool thing is you can sort of study the effects of these 2 separate regulatory processes if you have RNA dynamic information to show how these cells are related in time. And I think this sort of analysis is incredible. It's beautiful, and I'm really excited to see more work in studying these changing cell states. Okay. Okay, see, there are no questions. Great. So at some point, we actually thought about how to make this 4SU approach applicable to biological questions where you want to monitor multiple RNA dynamical processes. So you can imagine if we had a lot of different metabolic labels, you could pulse and chase in these different cycles many different times. Or even if you had 2, you could do like a dual pulse chase strategy, which you could do a lot more with in terms of your analyses. So Katharina Gusser and others developed this conversion of incorporated 6SG such that it's read out as A's in sequencing. The biggest barrier is that 6SG doesn't have a very high conversion rate. So after 1 hour incubation, they found that less than 5% of the transcripts were mutated. 6SG is much lower than 4SU's conversion rate, which is really high. They see about 11% conversion without the IAA, which they think is just because of some weird tautomeric base pairing. But if you add the IAA, which is part of the protocol, their conversion rate's over 94%. So this conversion rate needs to be really high, because if you're reading out your results in sequencing, you really want to get the most ability to read out all of this data and trust your what you're seeing for the spliced versus unspliced, or for the newly synthesized versus existing mRNA. So I think in general, tools for studying velocity are becoming more accessible and scalable, and I suspect that new mechanisms for metabolic labeling will improve our ability to study the transcriptional dynamics of RNA. Okay, so hopefully I've shown you a little bit about why being able to measure RNA velocity is useful. Conceptually, you can probably imagine that if you had many different metabolic labels or many different samples, you could start to estimate acceleration. And so here you can see maybe there's some ground truth of this transcriptional program. And as we sample more cells, we start to be able to better approximate the acceleration. But until we are able to have really refined sampling, at best we have an approximation of the velocity. So it's distinct from velocity in that you can get a prediction of future and past states, and many sort of complex transcriptional changes occurring in between. Another way of thinking about this is that you can get the information you want by measuring the age of every piece of mRNA that's transcribed by a promoter. So in this first example, maybe you have a cell that's trying to induce expression. So it turns on expression and then turns it off. In your age distribution, you have some curve like this, where the oldest mRNA was the first transcribed and the newest mRNA was the last to be transcribed. If the cell decides to induce expression at a different time point, the ages would be a distribution that would be shifted in the age and time. If a cell decides to turn expression on and off, and then turn it on at a later time point but less strongly, you would see a merged distribution of the ages of your mRNA. And then you can try to use what you measure in your age distributions to infer the program that might have been driving this expression. So in order to determine the age of each RNA, we developed RNA timestamps. And this project was driven by Sam and Lillian in our lab and uses an RNA timestamp as the substrate to record time. So this timestamp is an RNA that gradually accumulates A to I edits that are caused by the editor. And these edits are read out as A to G mutations in sequencing. So we designed the sequence to be very A-rich to maximize the number of editing sites and thereby our time resolution. Every editing site gives you another point to differentiate between the ages of two timestamps. And there are also these MS2 sites that serve as docking points for the editor in order to target the editing to the timestamps. The editor shown here is composed of this human ADAR2 fused to an MCP, or the MS2 capsid protein. And it's a protein that binds to MS2 sites in RNA. And so after screening multiple RNA and ADAR variants, we selected a pair that would give you an editing timescale in the order of hours. So while you can determine the relative age of RNA based on edits, we also wanted to calibrate our method to real time by determining the relationship between the number of edits that you see with real time. So you can imagine the way to do that would be to have the timestamps be expressed, measure the number of edits at various time points, and then build this relationship between edits and time. So we put the timestamp under the control of a tetracycline promoter, and in the 3' UTR of this IRFP gene, which shows minimal degradation in 12 hours. You don't really want your timestamps to degrade, because then you're introducing another variable and losing valuable data about your age distribution. And then afterwards, you co-transfect the RNA timestamp in the editor, and after 12 hours at time 0, add doxycycline to the media. So the expression of a timestamp reports the expression of this promoter, and the doxycycline induces the promoter. And so you let it induce for 1 hour, and then you stop all transcription with actinomycin D to further to inhibit any further downstream transcription that would affect your calibration curve. So then after that you lyse your cells at various time points to measure the amount of edits and build our curve. So then you can sequence your timestamps and see the number of edits at each time point. And you can see here that some sites are much more likely to be edited, but over time, the sites are gradually saturated. Here's a plot that shows the number of edits on the timestamp over time. And the number is highly reproducible between biological replicates. So you can model this as a Poisson binomial where each site has some probability of being edited, but each site can only be edited once and it can be saturated with time.
The question in the chat box reads, do timestamps impact RNA half-life post-transcription?
Okay, so the timestamp is part of, it's distinct from what this promoter would normally be transcribing. So if you were trying to measure the expression, or if you're trying to measure the age of let's say like your favorite gene, you would get the promoter for that gene and put it upstream of the timestamp on a separate construct. And so the idea is that what you're reading out with your timestamp is a good proxy for how many times you're transcribing your favorite gene. In terms of how, I guess, stable this timestamp is, we did do a test to see what the degradation was, and we found very little degradation in a 12-hour timestamp. Thanks, Jim. Cool. So thank you for that question. I think that's a really important nuance to pick up actually in the method. So, okay. So basically, you can, if you're trying to estimate the age of a timestamp, the lowest effort way to do it is just look at the number of edits and see how well it matches in your curve, right? And we found that you can estimate the age of each timestamp with a 95% confidence interval of 2.7 plus or minus 0.4 hours. So this plot shows the predicted time points based on different editing numbers. So that the x-axis is the number of edits, and the y is the inferred time point, and each column sums to 1. So the color is the probability that you're assigning it to that time point. As you can imagine, however, comparing to a single RNA timestamp is less effective than comparing to an ensemble of edits. The mean edits doesn't give you as much information as comparing to the entire distribution, which has a lot more information. And so the next step is, instead of just comparing one timestamp, what if we compared multiple and their distribution to the calibration curve? So the underlying transcriptional program is the function that determines the RNA expression. So maybe the cell will turn on transcription for a certain interval of time, leading to the production of RNA. The RNA made at the beginning of the interval will be the oldest and with the most edits, and the RNA made at the end will be the youngest with the least edits. And we're defining here transcriptional program as a set of 12 values representing 12 different hours that sum to 1, where each fractional value corresponds with the fraction of RNA that's made in that 1 hour window. So 1 here means that all of the RNA that we see is made in this 1 hour window at hour 2, for example. And so in our experiments, we observe an RNA distribution that's some subset of all the RNA in the cell. And using that observed distribution, you can then infer what the initial transcriptional program was. Another way to visualize this is in the form of a heat map, where each entry here is time and color is the probability that your transcriptional program, or the fraction of your highest probability estimate that belongs to a certain hour time point. So our algorithm for inferring this transcriptional program uses gradient descent to identify the convex combination of these various 1 hour time points that minimizes the L2 norm between the observed and the editing distribution associated with the combination. This combination is our transcriptional program. Intuitively, how accurately you can assign the time point that the RNA was made depends on how many RNAs you can sequence from a single time point. So to measure the accuracy, you can randomly sample RNA from one of your calibration single-hour editing distributions and see how often you correctly assign the time point. So here's a plot showing how often you can correctly assign the weight to a single time point given different numbers of RNAs that you're sampling from that time point. So as the amount of RNA goes up, your accuracy in assignment goes up. And this is the accuracy when you look at the assignment to a 3-hour window. Another way of looking at this is the amount of error for different ensemble sizes of RNAs. And so by looking at 4 RNAs, you're able to reduce your error to 1.49 plus or minus 0.2 hours from the original what I showed you before, which is just looking at a single RNA. When you look at 50 RNAs, you get a probability map that closely resembles what you would expect from the calibration curve. So what about more complex programs rather than a single-hour induction? So you can computationally generate these more complex transcriptional programs by making mixtures of RNAs drawn half from your 1 hour time point and half from the 5 hour time point. So applying the gradient descent algorithm, you can then infer this 2 peak transcriptional program. So here, the x-axis is the same as before, the number of RNAs that you're using to sample your sets, and the shading is the predicted time points. And with 10 RNAs, we're able to identify these 2 different peaks about 51% of the time. If you're able to sample 200 RNAs from this program, that increases to 80%, 86% of the time. So I think the simulation is a good sanity check, but it's also useful to do the experiment to see if what you get in real life matches what you would expect from the simulation. And so we tested this experimentally by transfecting cells with 2 different bar-coded RNAs. One was driven by a doxycycline-sensitive promoter, as before, as TET, and another by a light-sensitive transcription factor bibid. And so you can independently stimulate these cells with each promoter at 2 different times at 1 hour and 5 hours to get this editing distribution of 2 peaks. And then you can sequence your results and subsample different numbers to try to understand how your predicted time points improves with the number of RNAs that you're sequencing. So this is a similar plot to before, where you should be able to see 2 peaks. And this is a harder problem because when you're sampling, you're not constraining your sampling like you were in your computational simulation to equal numbers of each distribution. But what we actually got was that the number of RNAs, and if you control the number of RNAs in each set, you actually get pretty comparable percentages for how well you're predicting the 2 peaks. And here, let's say you're looking at 10 RNAs and you're sampling them like 100 times. This is the fraction of those samples that you can detect the 2 peaks, and that goes up with the amount of RNAs that you're looking at. So up until now, I think all the analyses that I've shown you for RNA timestamps have been in bulk. We also adapted this method to work in single cell data. We transfected cells with timestamped RNAs under the control of that same tetracycline doxycycline promoter, and then induced the cells according to 3 different single-hour induction protocols. So individual cells were sorted into wells, and then we performed single-cell timestamp sequencing. So here you can see the bulk distributions of the edits and some randomly picked single-cell distributions. Analysis of these editing histograms gives us the mean induction time associated with each cell. And this is the estimation on the error for single cells in these single-hour induction protocols. And for one of them, it was, the error was 0.2 plus or minus 0.8 hours, and another was 0.5 plus or minus 1 hour. And so the resolution on these shorter hour timescales is quite good. If you care more about relative time rather than exact time, you can imagine just ordering your predicted times from sort of lowest to highest time, right? And you can measure how many times what you would expect from your ground truth is switched to get the accuracy of your ranking. And in these cells, we only saw 5 transpositions, and that gave us an 85% accuracy in predicting the relative time. So you can also integrate these single cell analyses with 10x sequencing by performing an additional targeted amplification on the timestamps. This heat map on the right shows the differences between the predicted and the actual time points for the single cells. So each row is a single cell, and the x-axis is the number of edits, and the color is basically the fraction of each cell's editing distribution that belongs to a certain number of edits. So you can perform k-means clustering on the timestamps, and you get 3 distinct clusters that correspond to the 3 different induction points, where red is like 1 hour, light blue is 2 hours, and dark blue is 4 hours. And examining these cells, about 75% of the cells were assigned to the correct induction condition. And so the idea is then you can integrate your whole transcriptome analysis with a temporal element as well. So I think RNA timestamps is really well powered to reveal temporal dynamics in individual cells, but while it works pretty well for single hour editing, if you had a more complex transcriptional program where maybe you're slowly ramping up transcription over many hours, you'd have to have thousands of RNAs to be able to accurately infer that program. And in some systems, it might be a little bit harder to build this calibration curve, but of course, without the curve, you can still get these relative time relationships. So our lab is working on developing a mouse for in vivo experiments and multiplexing this analysis to multiple genes and to various other cellular events. But I think the takeaway here is that this idea of measuring programs and acceleration is a space that we'll see a lot of tool development in the coming years. So in conclusion, I think there are many new methods for RNA temporal dynamics that can help answer biological questions with changing cell states. And velocity-based methods are really useful for determining past and future cell states on the multiple hour scale. All these methods take the difference between an instantaneous measurement, which is the current cell state, and an integrated measurement building over the pulse. Acceleration-based methods can achieve sub-hour resolution and can uncover more complicated underlying transcriptional programs with the caveat that you might need to sequence a lot of RNAs. So this is a short acknowledgement slide, but I'd like to thank the Chen Lab for being so supportive and a wonderful place to grow as a scientist, and Sam and Lynn for mentoring me in my first project, RNA timestamps, in the lab. This was back when we were able to be together, and we went to an alpaca farm, which I think is kind of fun. As last point, I'll leave you with this. The third through sixth derivatives of time of position are jerk, snap, crackle, and pop. And I think as you go up in derivatives of time, it gets a little bit less intuitive, but it'll probably make for a really fun science paper to read. So if you're interested in the space, please pursue it. That's all. I'll take any questions if there are any, but thank you for the time that you took to participate.
Thank you so much, Sophia, for that amazing talk. I see that there is a question, but again, I can't read it. So there is a question in the Q&A.
I see. How are you specifically amplifying the timestamp signal before doing the RT-PCR? So I assume, oh, sorry about that. I assume you're sort of talking about here. You can have primers. Yeah, great, cool. Single cell sequencing. Thank you, Matthew. But you can have targeted primers because you know the sequences on your timestamps. So you would be able to sequence in the whole transcriptome, and then you would separately be able to also sequence with targeted primers your timestamp.
Thanks. don't see any other. I'm curious, in part just because of an experiment that I'm hoping to do in the future, whether you've applied this method at all to ask about sort of the time course of responses to a perturbation that is imposed upon cells at some point, in particular, one that maybe lasts a brief time and then watching its fallout as compared to one that is sustained over a longer interval. And this is kind of a general or vague question, but I wonder if you see any challenges or prospects for watching sort of the time course of a response to a brief as compared to sustained challenge, if you will.
Yeah, I think this is a space that a lot of people are interested in because, you know, neuronal firing happens really quickly, and maybe you want to measure how many times neuron is fired or something by looking at like CBOS or something, right? I think there are definitely some challenges to looking at very brief induction time points, just because you're getting much fewer RNAs, right? So it depends a little bit on how many RNAs you're getting, and if your time interval is really short, maybe that's not possible, but you can still integrate over a longer period of time your readout, right? So maybe you're having a lot of firing happening in like an hour time span, and then your timestamp readout can be have a lower time resolution but still be interesting. And I think we actually have a figure that tries some of this in the supplementaries if you want to look at that. But yeah, that's a space where people are really excited about.
Great, thank you. And I think there is one more question in the Q&A. So.
Sam says, great job, Sophia. Have you knocked in a timestamp into the 3 UTR of your favorite gene?
I have not, but hopefully one day I will. I think you know, with this timestamp mouse, hopefully everyone's favorite gene will be able to be analyzed one day. But this multiplexing it is actually a pretty big challenge, just because you can imagine you can't, like, there's a limit to how many timestamps the cell can produce. So if you wanted to study a lot of genes, then it's a little bit tough. But there are creative ways to try to record a time. You can try to, we thought about this at some point too, is just try to have an editor on the poly-A binding protein, and you can use the poly-A tail as a way to record edits and then read out your time. That way you don't have to worry about lots of different timestamps, but that's a whole other challenge in and of itself. So one day, I hope.